Welcome to this explainer. Right now, literally as you're watching this, there is a high-stakes, microscopic battle raging in fields and farms all across the globe. It's a war fought not with tanks or artillery, but with complex biology and genetics. On one side, we've got invisible armies of pathogens. And on the other, the crops that make up our entire global food supply. Today, we're going to unpack the fascinating science of how we protect the very food we rely on to survive. It's a pretty terrifying question, isn't it? What actually happens when a microscopic fungus or a super resilient virus successfully attacks our agriculture? Well, the reality is that plant diseases cause massive, catastrophic losses in agricultural production worldwide every single year. These pathogens aren't just chewing up a few leaves. They are a severe threat to global food security, human livelihoods, and our environment. Okay, let's get right into it by defining what we're actually talking about. When we say disease resistance in crops, we're talking about the innate ability of a plant to limit a pathogen's infection, growth, or damage. And this isn't just a simple shield. As we'll see, it's a highly complex combination of genetic programming, biochemical weaponry, and physiological defense mechanisms. Here is our roadmap for today's explainer. First, we'll look at the threat to our food. Then we'll explore how plants fight back, check out traditional crop breeding methods, jump into the biotech agricultural revolution, and finally, look ahead at the future of farming. All right, part one, the threat to our food and pathogens on the attack. To really understand the defenses, we first have to understand the sheer diversity and the relentless nature of these invisible armies. Our plants are up against four major categories of attackers. Fungi are responsible for some of the most devastating historic crop failures, causing diseases like rice blast, wheat rust, and the infamous potato late blight. Then you have aggressive bacterial pathogens causing wilts and blights. Viral pathogens, like the tobacco mosaic virus, completely hijack a plant's cells. And finally, nematodes. These are microscopic worms, like the root knot nematode, that silently destroy the plant's roots from underground. Moving on to section 2. How plants fight back with their fortress defenses. It's really easy to look at a plant and just think of it as a helpless victim sitting in the dirt, but that couldn't be further from the truth. Think of this like a mighty castle under siege. A plant's defense system is broadly split into two categories. First up is passive resistance. Think of these as the permanent castle walls, thick cell walls, waxy cuticle layers, tough bark, and little hair-like structures called trichomes that physically block invaders. But if those walls are breached, active resistance kicks in. This is the counterattack. The plant can trigger a hypersensitive response, which is essentially a rapid, localized cell death to literally starve the pathogen and stop it from spreading. It also deploys pathogenesis-related proteins like chitinases and powerful antimicrobial chemicals known as secondary metabolites to actively fight off the infection. Now, genetically speaking, this defense is structured in two different ways. Vertical resistance is controlled by single major resistance genes, or R genes. It provides an incredibly strong specific defense against one particular strain of a pathogen. However, because it relies on just one genetic lock, pathogens can sometimes overcome it. On the flip side, horizontal resistance is controlled by multiple genes. It doesn't offer total immunity, just partial resistance, but because it relies on this complex network of genes, it is highly durable over time. It's just much, much harder for a pathogen to adapt to a multifaceted defense. Section 3, traditional crop breeding methods, or human reinforcements. For thousands of years, humans haven't just watched this war happen from the sidelines. We've actively stepped in to help our crops build stronger defenses. Conventional breeding has always been our frontline strategy. The process is straightforward, but it requires immense patience. Step 1. Farmers and scientists identify plants in the wild or in fields that naturally survive an outbreak. Step 2. They crossbreed those natural survivors with our high-yielding, tasty agricultural varieties. Step 3. They carefully select the offspring that actually inherited the resistance. And finally, Step 4. After generations of testing, they develop a stable, improved variety that is ready for the farm. We're essentially playing genetic matchmaker to combine the best traits. But what if the resistance just isn't there in nature? Aside from just selecting what naturally exists, we've also used techniques like selection breeding and, fascinatingly, mutation breeding. When we lack natural resistance in a plant's gene pool, scientists have historically used gamma radiation or chemical mutagens to artificially create brand new genetic variations. The hope here is to randomly spark a mutation that provides the exact disease resistance we need. It's a pretty aggressive brute force approach to finding a cure, but it has worked. Which brings us to Section 4, the Biotech Agricultural Revolution. This is the tech upgrade. Today, we've completely shifted from that brute force approach to absolute pinpoint precision using a cutting-edge biotechnological arsenal. For example, we use marker-assisted selection to identify resistance genes in DNA, which speeds up breeding exponentially. We use genetic engineering to directly introduce resistance, like in bee crops that naturally deter insects. Then there are mind-blowing tools like RNA interference, which allows us to literally silence a pathogen's related genes, shutting it down from the inside out. We have CRISPR-Cas genome editing to precisely remove susceptibility genes from the plant, and genome-wide association studies to map out the exact genetic variations that create immunity. We are quite literally rewriting the rules of engagement here. To really grasp how precise this gets, we have to zoom way in to the molecular level. When pathogens try to infect a plant, they release specific molecules called effectors. You can think of these as stealth saboteurs trying to secretly unlock the plant's cells. But plants equipped with resistance, or R genes, have a high-tech alarm system built in. These R genes specifically recognize those pathogen effectors. The moment they detect the saboteur, they trip the alarm, instantly activating the plant's defense signaling and cell protection mechanisms. And finally, Section 5, the future of farming and winning the war. All this molecular biology is undeniably incredible, but you might be wondering, how is this actually impacting the food on our plates, the sustainability of our agriculture, and the future of our species? Well, this isn't just theoretical science. It is succeeding right now in the real world. We've successfully engineered blast resistance into rice, rust resistance into wheat, and late Blake resistance into potatoes. We've protected tomatoes from viruses, cotton from wilt, and maize from devastating fungal diseases. This translates to massive increases in food security across the world's most vital staple crops. Of course, we need to be realistic and look at the whole picture impartially. The advantages are just staggering. Higher productivity, massive reductions in chemical pesticide application, lower production costs, and far greater environmental safety. It is truly the backbone of sustainable farming. But there are limitations. Pathogens are living things, right? They continuously evolve new strains. Resistance genes can and do lose effectiveness over time. Plus, genetic modification requires immense time, huge resources, and faces steep regulatory challenges all over the world. Because of those challenges, scientists are already looking to the next horizon to stay ahead. The future involves finding broad-spectrum resistance genes that can protect against multiple diseases all at once. We're designing crops that are not just disease-resistant, but climate-resilient too. We're utilizing AI-assisted gene discovery to predict where the next resistance will come from, diving deep into synthetic biology, and even using microbiome-based protections, which basically means leveraging beneficial microbes to act as a living, breathing shield around the plant's roots. It's an incredible testament to human ingenuity. Through conventional breeding, genetic engineering, and CRISPR, we are basically building fortresses out of our crops. But biology is never static. It is a perpetual, never-ending evolutionary arms race. So it leaves us with this final vital thought to ponder. As we use our most advanced science to engineer the ultimate crops, are we truly ready for the new pathogens that will inevitably evolve to fight them?